All right, so let's get started, guys. Hi, everyone, and welcome. It's a great pleasure for me to introduce Dr. Andrei Irimia as our speaker today. Andrei is a professor of gerontology over at USC. So he calls himself a gerontologist these days. He has joint appointments in biomedical engineering, neuroscience, and computational biology. Andrei wears many hats. He has a MS in computer science and a PhD in biophysics from Vanderbilt. He then did his postdoc at UCSD first and then UCLA second at Lonnie, where we overlapped.
Yep.
And it was a great time. He then joined tech medicine over at USC, the Department of Neurology, as a junior faculty before being recruited to the USC Leonard Davis School of Gerontology, where he's now an assistant professor, but soon to be
associate already.
Yeah. Associate already.
Yeah, so yeah. Thank you.
So Andrei, his general research theme is in a broad topic of neural injury, and in that sense, he looks at brain imaging of TBI, neurovascular pathology, and neurodegeneration in atypical aging. Andrei is most well known for his award-winning approach for graphical representation and visualization of brain connectomics data. And hopefully I expect to see one or two slides on that here. So Andrei has used that approach for clinical use as well as population-based studies. And he's also used that approach to estimate the brain connectome for Phineas gauge and to map out the extent of the damage to his neural network. And I remember that was quite a buzz and excitement when you guys did that. Andrei these days is focused, so his research these days is focused on developing interpretable deep learning approaches for modeling human aging and has applications both to healthy human populations as well as patients in Alzheimer's disease. Andrei's had very recently, a few months ago, two very high profile papers in PNAS, which address this question. And so Andrei is going to talk to us about that and explain to us what he did and his latest research findings. So let's welcome Andrei.
All right. Thank you very much, Shantanu, for your very kind introduction, and thank you very much for having me here. As Shantanu said, I'm interested in sex-dependent anatomic patterns of brain aging that are specific to Alzheimer's disease, and I'm interested in resolving the puzzle of why there is such a marked discrepancy in Alzheimer's risk pertaining to sex. Females specifically are thought currently to be at high risk for Alzheimer's disease, and we have done some work on brain aging that tells us a little bit about that, and we have used deep neural networks to that end, partly because deep neural networks can allow us to find patterns of aging that are not apparent to the naked eye and to humans. So to explain the work that we did, I have to first describe the relationship and difference between chronological age and biological age. So we all have both. The chronological age is our calendar age. When we say of someone when we say that someone is 40 years old, we mean chronological age. However, we know that there is a lot of variability in the rates of aging across tissue types, across cell types, across people, and across organs. And so what researchers have found is that actually it's more useful to look at the biological age of a person to estimate a risk for disease. So if we look at the pace of aging, we see that the pace of aging is different in different populations. Let's see if I can. So you can see here in this graph, usually you have an average pace of aging in a healthy control population, but there are always people whose pace of aging is relatively slower or faster than expected. It might be slower, perhaps because there's less exposure to environmental factors that accelerate aging, or it could be that that person has a very healthy lifestyle or diet. Maybe they do more physical exercise. So that could be associated with a slower pace of aging. But it could also have a faster than average pace of aging. That could be due, for example, to excessive consumption of alcohol or obesity. It could be due to a sedentary lifestyle and or environmental exposures. It could also be that the genetic makeup of the person, given the environment, is not protective against aging. And so that could result in faster than average pace of aging. And so we see this heterogeneity in rates of aging across different organs, but also across different tissue types. And in this talk, I will focus on the brain. Chronological age is a single number. It doesn't describe the heterogeneity across people and across organs of aging. And so if we were able to quantify aging for each tissue type, for each organ, for each part of the brain, for example, that could give us better quantifiers of aging to estimate risk for disease, including, in this case, Alzheimer's disease. In the absence of any clinical indications, biological age is expected to equal chronological age on average across a population. And that's how we define biological age. We define it relative to a normative sample that we study. So for example, if we pick all 40-year-olds in a large population of, say, size 100,000, and we look at their average biological age, that biological age should be equal to the chronological age. And if we see individuals whose chronological age is 40, but their biological age is, say, 35, so then we would imagine that those people might be aging a little bit slower, and vice versa for those who are aging faster and whose biological age would be, say, 45 instead of 40. Even though all these people are of the same chronological age, they have vastly different risks for disease. And in addition to that, we have to also take into consideration sex differences that might affect risk for disease in addition to the risk to the pace of aging. And we have to consider the possibility that perhaps in certain environments or given a certain genetic background, there might be a marked sex difference in the pace of aging or in the risk of disease. So the age gap this is one of the names used for this measure, is the difference between biological age and chronological age. So again, with our example of a 40-year-old, let's say Anna is 40 years old chronologically, but let's say that her friends think that, oh, you know, she looks more closer to 30. What that really means is that we are associating a biological age that's younger than the chronological age that Anna has. And so on this plot of biological versus chronological age, Anna would be somewhere over here. So we have, on average, biological and chronological age being equal along this line, regression line. And then people who are younger than expected, who look younger than expected, given their chronological age, would be below this line. And people who look older than expected would be above the line. So this would be, these are the people we would expect to be at highest risk for disease. And these are the people who would expect to be at lower risk for disease. And so the difference between the chronological age and biological age is called the age gap. And if you have a negative age gap, you would be below this line. So that's what you would want. But if you have a greater than 0 age gap, then you'd be above this line. And so the age gap tells us whether aging is, whether we have aged more or less than expected given our chronological age. If our age gap is 0, then that would mean that we're aging at about the rate expected for someone of our chronological age. So this measure is really helpful. It can be translated into a lot of different areas, into a lot of different systems. You can look at the age of the cardiovascular system. You can look at heart age. You could look at the age of the arteries, of the blood vessels. You can look at brain age. You can look at the age of skin cells. So we know that sun exposure, cumulative sun exposure, is a risk factor for oxidative stress in skin cells, for the production of superoxide radicals, and for accelerated senescence of the skin. And so this is a concept that can be exported to a lot of different areas, and I'm going to focus on the brain because that's my area of work. And in brain research, there have been a number of brain age algorithms developed, and when they are applied to Alzheimer's patients, we find that if we look at the MRIs of Alzheimer's patients, their brains seem to be about 10 years older than those of cognitively normal adults. So what does that mean? It means that if you take, say, 1,000 controls and you use a neural network to guess or to guesstimate the biological age or the true age of these cognitively normal people, we would get on average a error of 0 because these are cognitively normal adults. But if you take 1,000 patients with Alzheimer's disease and estimate their ages from neural networks or some other method, we find that the age that the algorithm guesses is actually 10 years older than the true age of the patient. So that tells us that you can use MRI to estimate and to guess the biological age of an individual. And if the brain looks a lot older than expected, that's a reason to believe there is high risk for disease. So we have used deep neural networks to do this kind of work. You can use neural networks to look at an MRI volume, say of the brain in this case, and to estimate, guess the age of the person. The problem is that, as you may already know, most deep neural networks are black boxes in the sense that they are very complex. They have a lot of features that they rely on from the data to estimate the age of the person, but we don't really know what those features are, or they're not very interpretable. It's very hard to gain biological insight into what exactly the neural network is doing to estimate brain age. And so there have been a number of methods to gain interpretability and insight into what neural networks do to estimate ages of the brain so well. One of them is probing. So you could look at the inner logic of a neural network model and look at a certain feature that the neural network uses and ask how important that is. And this is tedious because usually these neural networks have a lot of layers, a lot of blocks, a lot of neurons. It may be that what a certain neuron is doing is very abstract, so that might not be very useful. Another way is perturbing. So if you change the MRI that we use as input to the network in some way, for example, you could remove a feature, you could remove a voxel in the brain and say, well, how does the answer change if we remove that voxel or if we change it? That could provide some insight, but that's also very tedious because there's so many features and so many voxels in these large MRI volumes, and it's very tedious to train a neural network over and over again while doing this kind of occlusion analysis, perturbing the data. You could also use surrogate data. So you could modify the MRI in a way that you think is convenient, and then see how the answer, the estimated age, changes. And that's also difficult to do because we don't always know how best to change the data to get the insight that we're looking for. So these are all tedious and not necessarily very useful ways to gain insight into what deep neural networks do that has to do with interpretability. So what we have done is we have, in collaboration with our colleagues in the School of Engineering, we've developed an interpretable deep neural network approach that can help us to identify brain areas and gain insight into sex differences and differences between cognitive statuses that pertain to interpretability of the deep neural network, and understand how aging acts on this neuroanatomic brain phenotype so that we can better estimate disease risk and brain aging rates for the purpose of stratifying patients. So we use these convolutional neural networks. Just very briefly, these are networks that look at various features of an image or an image volume in the case of MRI. So here's an example from a Nature paper that's very highly cited on how to identify a breed of dog. So you can decompose the image of the dog into RGB channels and then look at different features. You can look at the shape of the eyes or location of the eyes, or you can, as you can see here, in this, these are all representations for different layers in the network, but you decompose the image into ever simpler patterns and then let the neural network identify the patterns that are most important or most salient for performing the classification or estimation task that's been trained to do. And so this is intuitively how it works. In our case, what we have done is train the neural network to estimate what we call the saliency of the MRI volume. So in other words, we look at the weights of the network and how those weights change with at each voxel. So we can create the saliency maps that tell us how important that each voxel is to the neural network as it estimates the age of the person. So for example, in this workflow that's just schematic, we look at about 6,000 people from 4 different repositories, and we looked at their MRIs. We strip the skulls and just look at the voxels inside the brain. And then for each voxel inside the brain, we ask the question, is this voxel more or less salient to the neural network as it attempts to estimate age? And so we can, that allows us to create these subject-tailored maps that tell us the saliency of each person. So in one person, maybe the ventricular system here might be more salient. So maybe there is more white matter shrinkage that could entail this kind of ventricular enlargement. And maybe there's one specific area of the brain, like here in the frontal lobe, on in one of the hemispheres, which in that patient might be aging faster or might be more salient for determining age. So we get these patient-tailored maps of aging of the brain that also can be co-registered or interpreted in the context of the cognitive profile of the person. So if, for example, the memory of the person is bad, maybe the hippocampus or the temporal lobe might have higher saliency for determining age. And that's interesting information and potentially for a clinical setting as well. But we also see that there are differences between males and females that have to do with the rate of aging and the location and patterns of aging. So we can average these individual saliency maps across a lot of people, a lot of the two sexes, and we get something like this after appropriate normalization relative to head size and differences in neuroanatomy. And so we can average these after we do that, after we apply those corrections, and identify differences in sex that pertain to the aging of the brain. So for example, here in males, we see that there's more saliency in the ventricular system, which contains the cerebrospinal fluid, and in the frontal lobes, and somewhat in the insula. But overall, this is predicated on aging, predicated on enlargement of the ventricles. Whereas in females, we see a lot more in terms of atrophy of the gray matter. We still have ventricular enlargement as an overarching feature, but we also see that there are other areas of the brain that are more salient in females compared to men. And so here, if we do the difference between those two maps, we get map of the sex differences in aging. And again, we see more red in males, indicating that in males, the aging is more predominantly related to enlargement of the ventricles compared to females. And females, on the other hand, you have other areas of the brain and white matter. So this is the blue here, where you have more saliency in females. And those anatomic differences actually reflect the differences in cognitive aging that have been noted between males and females. So this was based on a deep neural network that we trained over 7,200 subjects. It was trained originally on these cognitively normal adults from ADNI, Human Connectome Project, UK Biobank, primarily. Most of the subjects came from the Biobank, but we also included subjects from other repositories to ensure generalizability of the deep neural network and to avoid overfitting the network to just one sample, which is actually a source of error in neural networks a lot of the time. So after we did that, we assigned the participants into a training set and a test set. And the training set was 80% of the sample, and the test set was about 20% of the total sample. And also, very importantly, when we trained the neural network, all subjects were cognitively normal. And then we used this neural network to look at Alzheimer's disease and differences in Alzheimer's related aging pertaining to cognitive domains. So as you remember at the beginning of my talk, I mentioned that the age gap is the difference between biological age and chronological age. So if the inferred brain age of the person, as inferred from this neural network, is equal or about equal to chronological age, the interpretation is that brain aging is about as expected. If the biological age is older than the chronological age, then you have faster than expected aging. And if it's younger than the chronological age, then you have slower than expected brain aging, which is what you would want. So this is the architecture of our network. The input is an MRI volume, T1 weighted MRI volume. And we have these five blocks here. And we have convolutional layers, max pooling layers. We have dropout layers in these over here to avoid overfitting the network to the data, to the training data. And we used a ReLU activation function to all the convolution and dense layers to make the output more sparse. So out of this, we obtained a feature map from which we calculated the saliencies for each subject at each voxel in the brain. And then these saliencies, these features are used to estimate the age of the person using regression. So it turns out that this architecture is really good at estimating age. And in fact, it's so good that it does better than any other algorithms that have been proposed or at least published up to this point in the field. So one benchmark of error is the mean absolute error in estimating age. So the MAE, mean absolute error, is the average difference, absolute difference between estimated age and true chronological age. And in a sample of control, that measures the accuracy of brain estimation very well. And so if you use just simple regression, you get on brain features, you get a mean absolute error of about 6 years. And these are some of the more popular methods that have been proposed up to this point. And this is our solution. And most recently, we actually improved on our architecture. And now we're down to an error of about 1 year on average, which is something we're writing up now. So in terms of percentage variance explained in age, our method also does best. And it explains over 96% of the variance in ages. And so again, it's doing better than most of the other methods that have been proposed. And this is a cross-sectional model where we're working on a longitudinal model that allows us to look at changes in aging or in the sites of aging in each person across time. So that's something we're working on. So for benchmarking, this method also does a lot better than others. We have, it explains more variance in the data, and the execution time is notably lower than that of other methods. We also have compared this to a method that a few years ago got a first prize in a contest challenge looking at different utilities of brain aging method calculations. And so this is doing better than all of these. And we reported this in PNAS earlier this year. This is here looking at performance parameters. And as expected, our method does better than all others that have been published, and the execution time is a lot shorter. And then we used this method to investigate differences in aging across the spectrum of cognitive impairment, especially particularly Alzheimer's disease and mild cognitive impairment. So this is the 3D CNN is our method. The SFCN is the best other method that we could find. It's the one that won first prize in this contest that I just mentioned. And as you can see here, the age gap, which is that difference between biological age and chronological age, is about 0 on average for both methods in the cognitively normal participants in the UK Biobank. Once you go to other samples, you also have approximately 0 averages for that difference in the CAMCAN sample, which is from Cambridge. And then in mild cognitive impairment and Alzheimer's disease, as expected, you have some differences that are larger than 0, partly because of the disease having this accelerated aging effect on the brain that causes the age gap to increase. But what's interesting here is that our method estimated the true extent of aging, of average aging, a lot better than the other method that we compared it to, that the second best performing method. And so we found that in the MCI sample in the ADNI repository, actually the average age gap is a little over 4 years. So on average, those participants with MCI have a brain that's about 4 years older than expected for people of their age. So this is for females. And then for males, we found similar patterns with, interestingly, a lower age gap in females in the MCI cohort compared to males. Although in the AD cohort, so these are people who have been diagnosed with AD, we have very comparable numbers in terms of the amount of aging that's been seen. Also, very importantly, this method captures domain-specific cognitive impairment much better than chronological age. And we have looked at AD-related neurocognitive endophenotypes. So this is a looking at, in A, we have comparison of biological age as computed using our method versus chronological age. And this is looking in cognitively normal adults. So in cognitively normal adults, where there's no AD pathology, we would expect the two methods to do about the same, because the reason for that is that you have chronological age and biological age being approximately equal on average. And so you see that there's hardly any differences between controls, between CA and BA for the cognitively normal. But when you go to C and D, which is the MCI and the Alzheimer's, or MCI or AD group, so this here we have MCI, we actually see that the correlation of biological age with each of these neurocognitive measures is considerably better than chronological age alone. And so that reflects, again, the fact that brain age is a much better measure, and it's much better at capturing domain-specific cognitive aging compared to chronological age. When we look at the combined group of patients with MCI or AD, again, this finding holds, and we see that whereas chronological age has barely any correlation with the cognitive scores, the biological age has considerably better correlation, and so captures that cognitive impairment a lot better. So I'd like to tell you a little bit about the saliency maps. The saliency maps allow us to identify parts of the brain that are being weighted more, that are more salient to the neural network as we estimate the age of the brain. So this is topographically organized, as you can see. It shows all the brain regions, and you have visual depiction of saliency. So this tells us what parts of the brain are most useful for estimating age. And this is an extension of a method that was initially proposed for a 2D case, and we extended it to the 3D case for the entire MRI volume. And basically the way this works is you obtain a score function across the 3D volume for the CNN, and that score function tells you how each voxel is being weighted by the neural network when estimating age. And then those voxels are ranked based on their importance. And so if you have higher importance, then if the voxels have higher importance, then you get higher saliency. So you have you see these brighter areas in the ventricles and also in the temporal lobes as expected. You see here in the axial view, more saliency in males in the ventricles. In the females, not so much in the ventricles, although you can see it, but you find a lot more in areas here in the frontal lobe and more in the gray matter. So this is the saliency map actually allows us to quantify aging at certain parts in the brain that are very specific to patients or to a certain sex. And so you see here, again, males' brain aging is more strongly predicated on the white atrophy of the white matter and on the enlargement of the ventricles. But in females, there's a lot more involvement from the frontal lobe. And so you have frontal lobe atrophy of the gray matter that's being weighted a lot more. And so these are in slice cross-section differences in aging between males and females in cognitively normal adults. And red are areas that are aging faster in males, and blue are areas that are aging faster in females. So you can see there's a very pronounced pattern. You have more diffuse white matter aging in females here, at least in these areas towards the back of the brain, in parietal lobe, occipital lobe, areas that are usually also metabolic on PET scans in Alzheimer's disease. And then in males, you have a higher saliency in the ventricular areas where you have again, this ventricular enlargement feature. So we can also plot these differences in aging on the cerebral cortex. And we find that, again, red represents males aging being more salient than females aging, and blue is the other way around. So we see that, first of all, there is a pronounced asymmetry relative to the right or left hemisphere. So there's a lot of lateralization in aging patterns across the sexes. And we compared this to the lateralization literature on cognitive decline and cognitive impairment. And what we found is actually that these findings match what the literature has found very well. You have in males aging that's more pronounced in the cingulate, temporal, and dorsolateral frontal lobes. And because most of these subjects were right-handed, this data favored the scenario where, you know, the saying, if you don't use it, you lose it. In other words, but particularly as this applies to men. So in other words, what I'm trying to say is that the subdominant hemisphere in men is aging faster than in females. And female saliencies, on the other hand, are higher in areas that are more involved in AD pathology, like occipital, parietal, and some frontal regions. And this is, again, consistent with the findings that females have higher risk for Alzheimer's disease. Also, males' brain aging more often affects certain areas that are morphometry related, like crowns of frontolateral gyri. So the crowns of the gyri in these areas are aging faster in males, but in females, it's actually the troughs of the sulci. So it's at the bottom of the grooves, of the cerebral grooves you see here. So all of these findings had already been reported in the literature. These are not new findings, but we actually had to do quite a bit of a literature review to identify this and put them together, because the findings on differences in morphometry between the two sexes are scattered across the literature. And this is the first map of the brain that actually depicts differences in aging across the two sexes. And so this actually, even though it's not entirely new per se, it synergizes a lot of the literature findings that were not previously described with this kind of focus. We also see differences between cognitively normal adults and Alzheimer's disease. And we find, for example, here's the difference map between what you see on the left and in the center. So CN is cognitively normal, CI is cognitive impairment, namely with Alzheimer's. And if we look at the difference of these two maps, we find a lot of saliency that's more pronounced in cognitively impaired adults, so that's the red, in areas of the brain stem where AD pathology is first seen, which is places like the locus ceruleus and other nuclei of the brainstem, where postmortem histopath findings are that that's where the AD pathology is first seen. We also see it in the cerebellum. And so there's all these interesting findings that sort of make sense and are able to summarize in a visual way a lot of disparate findings in the literature on AD-related aging. And this is the plot on the cerebral cortex of the plots you saw in the previous slide. And again, we have some lateralization effect. And in cognitive impairment adults, we see aging a lot faster in cingulate areas and in the temporal lobe, on the ventromedial aspect of the temporal lobe, as expected in Alzheimer's. Here's an example from another population. So this could be used to estimate aging in a lot of different populations. And this is from patients with mild traumatic brain injury, where it's very important to get a measure of local and patient-tailored aging, because every subject is different, every injury is different. So you want to know at the patient level what the brain aging profile is. So this is looking at the homunculus and the primary somatosensory cortex, which actually this was revised very recently in a paper that was published in Nature. So this is the old version. But what's interesting here, at any rate, in mild TBI, is that if you look at differences between TBI and healthy controls who were matched to the sample we looked at, which had about 150 people with mild TBI. If you look at differences in aging saliency, a lot of these differences, interestingly, are in this area of the homunculus that are associated with head and neck somatosensation. So in the places where the somatosensory cortex neurons are processing maybe pain associated to TBI, or where you have maybe higher levels of oxidative stress in neurons that are processing information related to injury, you also seem to be having higher saliency that's potentially associated with faster aging of those brain areas. So this is, I find this to be very interesting, and we're planning on studying this further, but this is only to give an example of how you can have different patterns of aging according to clinical condition, and that might be representative of certain phenomena that occur at the population level, but you could also look at individual levels, and as expected, in that case, we also saw that there's more aging after a concussion in areas of the brain that were exposed and more impacted by the blunt force of injury. And so you have that kind of finding that draws a parallel between area of injury and accelerated aging of the brain. So this was published earlier this year in PNAS, and we had some exposure in the media. I encourage you to look at this paper if you're interested in the details of this work. The reference is Yin et al. In PNAS 2023. We have the software for calculating brain ages available on our GitHub, so please feel free to take a look. If you'd like to use this software on your own data, we'd be happy to help you, and we're actually very interested in looking for new collaborators who are wanting to apply this to new data sets or other data sets. We're very interested in collaboration. And we ourselves have some funding from the NIA to continue this work as part of an R01 for the next 5 years. I'd like to thank you for having me here, and I'd like to thank the NIH, the DoD and the Femino Foundation for funding this work. Thank you very much, and I'd be happy to take any questions. Thank you.
So excellent talk, Andrei. And
Thank you.
I remember the previous approaches using linear regression.
Yeah.
And this has come a long way.
Right, yeah.
A lot more.
Yeah, so the linear regression just does maybe you can get, in cognitively normal adults, you can get about 6 years MAE, and we're down to 1 year. So, yeah.
Any questions?
Yes. Curious if you apply software to any other modalities. Yeah, so we're interested in extending this to CT, which would be useful for people who are not getting MRIs or cannot get MRIs. You have to train another network, obviously. I think the challenge is that because you need so many subjects, you have to find enough data from controls. And it's not very frequent that controls get CTs of the head. So for CT, it might be hard. For other methods, there have been people looking at FLAIR or including with T2 or combining T1 and T2 and FLAIR and all kinds of ASL and other methods to improve on the age estimation, or to find new insights that maybe, for example, in ASL, maybe you have aging of the arteries or veins or just the vasculature that tells you something about aging. So that could be applied to that. We haven't tried it. We haven't found sample that's large enough for ASL. We could do this on T2, so we thought about that. Right now, this is applied to T1. And then again, there's a lot of people interested in using fMRI to find this, to estimate a functional brain age. So we're working on that project with a collaborator. Yep.
So meanwhile, the saliency maps, can we think of them as feature importance?
Right. Yeah. Yes. Yes.
And so my question was how to, and I'm sure you've done this, but maybe help me understand how to avoid the circular argument of selecting a feature in terms of its importance and then correlating it. And I'm guessing, so, and tell me how is this feature importance calculated? So do you have age and other age associated features that actually go into estimating saliency? And if yes, when you're trying to correlate it with other domains, should we expect that, yes, it will correlate?
No. So the way it works is that you produce this map that's a linear combination, or actually nonlinear combination of all the weights in the network. And that map tells you how each voxel is being weighted towards estimating age. And if the estimate, if for one voxel, maybe say it's 5, but for another one, it's 0.5, then the one with the higher weight is more salient. So this is salience in terms of the estimation of age. So that we don't pick the features a priori. The neural network picks the feature. Yeah. Yeah. Yeah.
Unsupervised? Can I
say well, it's supervised in the sense that yes, yeah. So you have the you're training the network to find what parts of the brain, what voxels are optimal for explaining for predicting the chronological age of the person. Yeah, so it is trained. Yeah.
Just trying to see if there are questions on chat. So then I had one more question. The brain aging score. I don't know, you didn't talk about a score. Maybe you have assigned a score, obviously, that comes out from this neural network.
So the neural network outputs an age.
Yes.
And it just outputs an age. And we know the true age. And so we're looking at the difference. And so that's what is the greatest insight. Yeah. Yeah. Yeah.
So my question is, how do we separate the brain aging part from the neurodegeneration or trauma or these other things that affect the brain? So how?
Yeah, so the way to do that is to just do a group comparison between the saliency maps of controls and the saliency maps of the group of interest or subject of interest. Yeah.
And you will get differences at different sites.
Yes, at different locations in the brain. And in fact, you could also look at controls. Let's say, where is my brain? If you want to ask the question, where is my brain aging more? Well, you could do a Z map, like kind of a Z-score map between your brain and the distribution of cognitively normal adults of your age and sex. And then you can say, well, this part of the brain, in your case, is aging a little bit slower than you would expect, which is great. But it could also be aging faster. And then you can get to see, where are you relative to others of your age and sex? Is your brain aging faster or slower than expected? And where is it aging faster or slower than expected? Is it in the temporal lobe? Is it in the insula? Is it the hippocampus? And what we've found is that actually the answer maps onto the cognitive performance of the person whose map and analysis you're doing. Yeah.
So that was the neuroendophenotype graph that you showed.
Right. So that was looking across.
Cognitively normal.
Right. Cognitively normal, and also where biological age and chronological age did very similarly, because you don't expect them to be different. But in cases like AD, where you have a big difference between biological age and chronological age, you get way better correlation of cognition with biological age, but almost none with chronological age. Yeah.
And let me just check if there is any other
question. Yeah.
That was the paper or elsewhere explore the behavioral,
So we didn't explore, so this was cross-sectional, so we didn't look at, we only looked at cognitive measures and neurocognitive assessments and AD and tests used for AD diagnosis. But we do know that actually from the in the MCI sample we used, which was about 300 to 400 people, about 54 converted to AD within 10 years. So we want to now look at longitudinal changes in aging and also changes in aging in response to interventions, like lifestyle intervention. And there could be things that are senoprotective, so protective against aging, and maybe, for example, exercise, an exercise routine might lead to rejuvenation of the brain, or becoming, say, very sedentary during the pandemic could be related to accelerated aging of the brain. Yeah.
Okay. There was a question from the audience. Can brain age be used to investigate the efficacy of therapeutic intervention? And it said, never mind, it will just answer, but if you want to...
Yeah, so yes, so I think it totally can. I think the best, this model is actually only for cross-sectional data. But if you have a longitudinal data set, or if anyone in the online or here has a longitudinal data set, would be interested in collaborating, because we have we've been working on this longitudinal model that looks at at changes in brain age. So here's and so then we could look at saliency at time point 1, saliency at time point 2, and here are the areas that have aged faster or been rejuvenated, or maybe they've stayed the same, but you can map that across the cortex. So we're interested in that.
So then I have one last question. The brain age, which is the predicted output of the deep neural network, is kind of a composite of many different features.
Right.
In a different sites. So even if it's a single number, I mean, have you thought of kind of decomposing that number, for lack of a better word,
yeah,
into and you already have these saliency maps to begin with, so
yeah, so you could look at, for example, age of the parietal lobe, or age of the insula, age of the hippocampus. So you could create, your own ROI and say, what's the age of my ROI? And then look at the ROI over subjects. And so we could do that too, yeah.
So currently, does the network output different ages, or you get a single age?
You get a single age, but you could, I mean, it's really... Yeah, well, actually, I don't think you'd need to retrain it. I think you just need to, yeah, I mean, well, I think because you usually it's not the age itself that's interesting. It's usually that age gap difference from chronological age. And so for that, you can just do some post hoc analysis to calculate that, yeah, from the data, from the whole brain data. No.
Great. So
okay.
So let's thank Andrei again for such a wonderful talk.
Thank you.
This is the end of the seminar, and see you guys next time.
Okay. Thank you.